So to discuss both the phosphagen system and anaerobic glycolysis, what we did is we focused on like a 100-meter dash, a sprint. In something like a 100-meter dash, those two systems are going to be the dominant systems. Towards the end of the sprint, you're going to see fatigue because the phosphagen system runs out, and anaerobic glycolysis cannot keep up with ATP demand. Without the phosphagen system, the idea is that anaerobic glycolysis on its own can't keep up with the ATP demand, especially of the faster twitch fibers, as we'll talk about later on in the semester. And if ATP demand is higher than supply due to the phosphagen system being depleted towards the end of the sprint, with a higher ATP demand than supply, ADP, inorganic phosphate, and also hydrogen ions actually, we get hydrogen ions from the hydrolysis or breakdown of ATP. Those molecules begin to accumulate in the fibers. So ADP, inorganic phosphate, hydrogen ions, right? Those molecules begin to accumulate towards the end of the sprint inside of especially the faster twitch fibers, and we begin to see fatigue. And if you were to try to continue this sprint on indefinitely, you can be 100% certain you're going to get total fatigue. No one on Earth can continue on with a sprint indefinitely. The muscle fibers will fatigue. Now what we're going to begin to do is talk about other systems. We're going to start off with this other system that is traditionally called aerobic glycolysis, the breakdown of glucose with oxygen. And understanding aerobic glycolysis, the breakdown of glucose with oxygen, will eventually actually allow you to understand how anaerobic glycolysis, this process of breaking glucose down without oxygen, occurs, at least according to this oxygen theory that I am assuming is fact in this class. Okay, so to talk about aerobic or slow glycolysis, the first thing that we need to do is think of an instance where ATP demand is not extremely high, but also not low either. Something kind of in the middle. We'd say like a moderate intensity. The best thing to think of, for me as an example, might be like 6 miles an hour or 7 miles an hour on a treadmill. For you, that's probably very, very different. It's maybe worse, it may be better. I don't know. It's going to be highly specific to the person. Think of them, again, just an instance of kind of moderate intensity. ATP demand is moderate. It's certainly not extremely high like it was in our sprint. In that instance, glycolysis is going to be running for all of the same reasons that it was before. If the muscle fiber has like a moderate demand for ATP, that means it's consuming a moderate amount of ATP, and what's going to be accumulating to a degree inside of the muscle cell. Oops. ADP is going to be accumulating inside of the muscle cell. Oops, there we go. Don't know what's going on with my screen there. So ADP accumulates inside of the muscle cell to a degree. What that's going to do is the same things as before. Yes, it's going to trigger the phosphagen system to a degree. That's something that throws people off because they think that these systems are like mutually exclusive, and they're often taught that way for simplicity. And I'm going to continue to actually teach it that way. But really all of these systems work together. It's just more so a question of which ones dominate in which circumstances and why. So during this moderate ATP demand instance, moderate amounts of ATP will be utilized and ADP will accumulate to a degree, and it will trigger the phosphagen system to be active. Like, creatine kinase is activated by ADP. Phosphocreatine is activated and stimulated by ATP to a higher degree than it normally is. So it's not like the phosphagen system wouldn't be utilized to some small degree here. But the important part is it's the other systems that are going to dominate in this instance and are most important, not the phosphagen system. Because ATP demand is not that excessively high here. So
I want to mention that about the phosphagen system, but I also want you to kind of try to get that out of your head. ADP is triggering glycolysis also to run, right? It's activating phosphofructokinase, so glycolysis is running, calcium's accumulating inside of the muscle fiber, epinephrine is rising in the bloodstream, moderate ATP demand instance, right? 6-7 miles on a treadmill. Epinephrine in the bloodstream accumulating, calcium in the muscle fiber accumulating is also triggering glycogen. I can write that over here, right? Glycogen to be broken down into glucose, specifically glucose 6-phosphate. ADP is triggering glycolysis to run at a faster rate. So we're running glycolysis during this moderate ATP demand instance inside of the muscle fiber. Okay. Glucose ultimately here is being broken down into pyruvate. So we have pyruvate and we have NADH that's being produced, both of these things. And glycolysis is also, of course, producing ATP as well, as you know. But the important thing here is when ATP demand is more moderate, what's going to happen to pyruvate and NADH is completely different from our last example. So that's what we need to discuss now. So you know that when ATP demand is very high, a lot of the pyruvate that's produced, not all of it, but a lot of it, gets converted into lactate, just write L there, to regenerate NAD to keep glycolysis running at a rapid rate. And the driver behind that is most likely due to insufficient oxygen. Okay, other possibilities too. But that's what we're going to focus on. So, if ATP demand is more moderate, not super high like during the sprint when most of pyruvate is converted into lactate, with a more moderate ATP demand, what we believe, one solid theory, but there are others too, is that oxygen levels are more sufficient. That's probably the best word to use for this. And with more sufficient oxygen levels, what's going to happen is that pyruvate, as well as the NADH, instead of the reaction that occurs when ATP demand is very, very high and oxygen is insufficient, where pyruvate gets converted into lactate to regenerate NAD, in this instance, when ATP demand is more moderate and there's more sufficient oxygen levels, the pyruvate, as well as the NADH, through a pretty interesting mechanism, is going to get shuttled into the mitochondria inside of the skeletal muscle cell. Most of it, I should again emphasize, is going to get shuttled into the mitochondria rather than pyruvate being converted into lactate and NADH being converted back to NAD. So now the whole point of this discussion is really to discuss why, in this instance, pyruvate and NADH get shuttled into the mitochondria when ATP demand is more moderate and oxygen is more sufficient. So before we go any further, I want to talk about the anatomy of the mitochondria. They all have an outer membrane, which I'm trying to do my best to highlight. There's the outer membrane. And this would be all-encompassing. It's been cut in order to show you the inside of the mitochondria as well. So there's the outer membrane, which encompasses the whole outside of the mitochondria. It's kind of like the plasma membrane of a cell. And then there's an inner membrane. This right here is the inner membrane. And it exists, I'll just, there are these folds all over the place. This is all the inner membrane. Okay? So there's an outer membrane and an inner membrane. In between the outer and the inner membrane is what's called the intermembrane space. You know what? I should use a different color for that. I'll use it in red. In red, there's the intermembrane space. This is all the intermembrane space, and it would be in here too. There's the intermembrane space, the space between the outer and the inner membrane. Okay. Deep to the inner membrane, I'll do it in green, is what's called the matrix of the mitochondria. So you have to get past the inner membrane to be in the matrix. The goal of getting molecules, or I shouldn't say this, when I say molecules go into the mitochondria, what I mean is they get into the matrix, because that's where all the important stuff happens. So collectively, what do you need to do? You need to get molecules past this outer membrane, then past the inner membrane into the matrix here. So we have to get molecules through two membranes. Generally, it's very easy to get molecules through the outer membrane. The outer membrane is very leaky. There's lots of pores through it. Molecules go through it quite easily. There are exceptions to that, and we're actually going to talk about one pretty quick here. But generally, most molecules can get through the outer membrane. It's the inner membrane that they don't have as easy of time of getting through. There's lots of what are called integral protein, transport proteins, in order to get molecules from what's known as the inner membrane space here. So molecules will get through the outer membrane into the inner membrane space, and then they need help getting past the inner membrane and into this matrix here. Okay. So I'm going through all of this because when I say, most of the time, when I say a molecule is being transported into the mitochondria, I'm referencing the molecule traveling past the inner membrane into the matrix. Oftentimes, people just don't even discuss the fact that the molecule travels through the outer membrane. When they talk about how the molecule is transferred in to the mitochondria, they're talking about the molecule getting into the matrix. They're talking about it getting past the inner membrane and into the matrix, and they're just disregarding the fact that the molecule is going through the outer membrane too, because it's so easy for that molecule to go through the outer membrane that they don't even bother discussing that component of it. They're just talking about how the molecule gets past the inner membrane into the matrix. So to pick up where we just left off, in this diagram, what it's doing is it's showing molecules being basically shuttled into the mitochondria. You can see these different molecules being shuttled in to the mitochondria. What I really wanted to point out before I get into discussing any of that is that mostly when a diagram shows molecules being shuttled into the mitochondria, it just shows the inner membrane of the mitochondria and shows those molecules being shuttled past the inner membrane into the mitochondria. The space beyond the inner membrane is called the matrix. And then it doesn't really show the outer membrane at all. The outer membrane of the mitochondria would be right about here. And between the inner and outer membrane is what's called the inner membrane space. So everything in here is the inner membrane space. And you can see that these molecules have to go basically from outside of the outer membrane, past the outer membrane, into the inner membrane space, before they can get shuttled into the matrix. Okay, so that makes sense, right? You got to get molecules past the outer membrane, into the inner membrane space, and then past the inner membrane into the matrix. And you look at these diagrams, and what you think of, at least if you're me, is why is there no depiction of the outer membrane? And why are they just referring to anything outside of the inner membrane to be the cytosol? Because the cytosol in biology is just the fluid inside of our cells. It's the intracellular fluid. Anything that's inside of an organelle, like this particular mitochondrion, the fluid inside of it is not considered to be the cytosol because it's fluid inside of a particular organelle. Intracellular fluid, the fluid inside of our cells that the organelles float around in, is the cytosol. Okay, so this fluid right here is technically in the inner membrane space inside of any mitochondria. So it's really fluid inside of an organelle. So why are they just calling it the cytosol, which should be everything that's outside of the outer membrane, outside of the organelle, the mitochondria? It's not differentiating between the fluid that's in the inner membrane space and the fluid that's entirely outside of the mitochondria. You know, if this were me, I would think of everything out here as being the cytosol. And in here, this is really fluid of the inner membrane space, which is mitochondrial fluid. But this diagram, and many of them, don't differentiate between the fluid in the inner membrane space and the fluid outside of the outer membrane at all. And the reason that they don't really differentiate is because molecules so easily can travel past the outer membrane, and I mean travel past it in either direction, right? They can go into the inner membrane and out of the inner membrane pretty much equally as easy. That they just, many diagrams, they just don't differentiate between the fluid at all. They just call anything outside of the inner membrane the cytosol. I would argue with that to a degree, but that's why you see it this way. And that's why I wanted to emphasize how leaky the outer membrane is. It's so leaky to most molecules, and I'm about to actually show you an exception, but it's so leaky to most molecules that a lot of text and diagrams don't even differentiate between the fluid that's in the inner membrane space and the fluid that's outside of the outer membrane. So back to the important part of this, you're seeing different molecules being shuttled into the mitochondrial matrix. Okay? You're seeing pyruvate being shuttled in, and when pyruvate is shuttled in, this is called an antiport. It gets shuttled in exchange for a hydroxide ion going in the opposite direction. Here you can see phosphate, inorganic phosphate, being shuttled in to the matrix and also in exchange for a hydroxide ion. And here you can see ADP being shuttled in to the matrix in exchange for ATP. In order to operate aerobic or slow glycolysis effectively, we want all of these molecules that are out in the cytosol to be brought into the matrix of the mitochondria. I already told you in lower ATP demand instances, when oxygen is more sufficient, pyruvate gets shuttled into the mitochondria, and that's what this diagram is showing you. And again, it's just showing it going past the inner membrane, but it goes past the outer membrane. It's to pyruvate. Pyruvate does, excuse me. And also, you've got inorganic phosphate going in, and you've got ADP going in. You want both of those also to be shuttled into the matrix as well, because they're going to be used in this process to resynthesize ATP. Okay, so at least now you can see how pyruvate and others end up in the matrix of the mitochondria. Now we need to talk about NADH.
So before I move on to NADH, I actually want to show one more thing. So you might, just to clarify, you might be thinking, well, where is this ADP and inorganic phosphate, ADP, where's it coming from? Well, remember, in this moderate intensity instance where ATP demand is moderately high, 5-6 miles an hour on the treadmill, the muscle fiber is breaking a good amount of ATP down, right? And when ATP is broken down, it's hydrolyzed, it's turned into ADP and inorganic phosphate. Now, some of that ADP and inorganic phosphate is being resynthesized in glycolysis, but a good majority of it, And again, not all this can make sense until you get through everything, but a good majority of it, when ATP demand, and these things go hand in hand, when ATP demand isn't that high, and when oxygen coming to the muscle fiber in theory is sufficient, okay, when these two circumstances are being met, not as high of an ATP demand and with more sufficient oxygen, a good amount of, a really good amount of the ADP and inorganic phosphate that's being created is actually being shuttled into the matrix of the mitochondria. So that's where this ADP and inorganic phosphate that's going into the matrix of the mitochondria is coming from, first of all. And the reason that it's going in there, the majority of it, is because of the circumstance that we're talking about right now. Now, what you'll see is that ATP demand, as ATP demand gets really, really high, the ability to bring ADP and inorganic phosphate theoretically into the matrix of the mitochondria, it's not that it becomes diminished, it's that the capacity to bring ADP and inorganic phosphate into the matrix of the mitochondria will eventually just get sort of maxed out, and ADP and inorganic phosphate will really kind of accumulate and get trapped out in the cytosol. Again, I don't expect that to make a ton of sense right now, but that's where all of this ultimately is headed. All I want to emphasize for the moment is that in the circumstance that I'm talking about right now, with more moderate intensity exercise, ATP demand isn't nearly as high inside of the muscle fiber as it was during the sprint for, I think, obvious reasons. And with a lesser ATP demand and more sufficient oxygen supply to the muscle fiber, which those two things go hand in hand, the good majority of ADP and inorganic phosphate that's created is all getting shuttled into the matrix of the mitochondria. Now, what about the NADH that's created in glycolysis? There's so much in this class. This is another one of these pathways that adds little to your overall knowledge of metabolism. So I'm going to bypass it for the most part. But what I can tell you is the mitochondria, the muscle fibers, are incapable of actually shuttling NADH itself, the NADH that exists in the cytosol, in the intracellular fluid. It's incapable of actually shuttling that NADH past the inner membrane of the mitochondria. So what's used is what's called the malate aspartate shuttle. Basically, NADH donates its hydrogens to something else, another molecule, and then that molecule takes the hydrogens into the matrix of the mitochondria, and then that molecule that takes the hydrogens into the matrix of the mitochondria gives the hydrogens back to an NAD molecule in the matrix of the mitochondria. Again, this is specifically NAD that exists in the matrix, not NAD that exists in the cytosol. Okay, this is NADH in the matrix. This molecule gives those hydrogens back to a molecule of NAD in the matrix of the mitochondria, and thus NADH is created. If you want to know the specifics of this pathway, again, a great idea to take a more advanced course. Why don't we simplify and just say this? One way or another, that NADH that got created in glycolysis, although not directly, that NADH, specifically the hydrogens from that NADH that was created in glycolysis, ends up in the matrix of the mitochondria as a molecule of NADH. So sort of indirectly, we get that molecule of NADH. Actually, we got two of them, two NADHs from glycolysis. We get those two NADHs from glycolysis into the matrix of the mitochondria, be it in an indirect way.
I wanted to provide you this image for clarity too. On the left, this is one muscle fiber. Inside of the muscle fiber, you can see the actual mitochondria. And hopefully you can envision ADP, inorganic phosphate, pyruvate, NADH, although that's a different mechanism, but all of these are headed into the mitochondria. Different mitochondria. Like there's lots of pyruvate, NADH, inorganic phosphate, and ADP being produced all throughout the cell, and they're going into different mitochondria. Okay, so hopefully it's helpful to see the mitochondria inside of the muscle cell and trying to imagine it, being produced through the breakdown of ADP and inorganic phosphate, and through glycolysis, pyruvate and NADH occurring in the cytosol, as it's called, in the intracellular fluid, and then making its way into the mitochondria.
And this also seems like a great time to point this out. If you look at a skeletal muscle fiber, this is one skeletal muscle fiber out of this whole muscle, right? This is one skeletal muscle fiber. And the mitochondria, okay, the mitochondria inside of a muscle fiber, there's actually two different populations. There's what's called subsarcolemmal. Those are the mitochondria that exist right underneath the sarcolemma, which is the plasma membrane of the muscle fiber. And then there are also what are called intermyofibrillar mitochondria. These are the mitochondria that exist in between the myofibrils, the kind of where the sarcomeres are, the contractile units of the muscle fiber. There's a potential question on the actual comprehensive exam related to this, so I wanted to mention it here. This isn't necessarily hugely relevant for this course, but it is a topic of research, the differences between these two different populations of mitochondria. They adapt very differently during exercise. Their structure and shape is slightly different. There's all sorts of interesting things about these two different populations of mitochondria. And again, in a more advanced class, it's certainly something that we could talk about a good amount after we understand the overall function of the mitochondria, which is where we're headed now.
So here is this image of pyruvate being shuttled into the mitochondria. And you think, what happens to pyruvate once it's in the mitochondria? Well, pyruvate gets converted by an enzyme called pyruvate dehydrogenase into acetyl-CoA. Before I say anything about what happens and how we convert pyruvate to acetyl-CoA, I want to mention when we broke down glucose in glycolysis, remember we got 2 pyruvates, right? So we're going to turn 2 pyruvate molecules each into an acetyl-CoA. So this isn't just happening once, this is happening twice. 2 pyruvate molecules are made in glycolysis. Okay, so how do we turn pyruvate into ultimately acetyl-CoA? Well, this is an oxidation. Pyruvate is being oxidized. Look at this. NAD comes along and it stole some hydrogens off of pyruvate and this created NADH. Okay, so we stripped some hydrogens off of pyruvate to make acetyl-CoA, and that of course happened twice. So we get 2 NADH molecules. Additionally, in this process, we stripped a carbon and 2 oxygens off of pyruvate. So we also created CO2, carbon dioxide, a byproduct. Right? This happened twice because we turned 2 pyruvates each into an acetyl-CoA. So we also got 2 CO2 molecules from breaking down each of these pyruvates into 2 acetyl-CoAs. Also, to turn pyruvate into acetyl-CoA, a coenzyme was added to pyruvate to make acetyl-CoA. So all of these things happen when we convert pyruvate into acetyl-CoA. If there's a big thing to take out of this, it's the fact that pyruvate is being oxidized, right, by NAD. NAD is taking hydrogens from pyruvate to make acetyl-CoA, and now we have 2 more NADH molecules. Just wanted to mention this since I didn't say it explicitly. The enzyme that's involved in converting pyruvate into acetyl-CoA is pyruvate dehydrogenase. If you remember, lactate dehydrogenase converts pyruvate into lactate. Pyruvate dehydrogenase converts pyruvate into acetyl-CoA. Now, of course, the question is, what happens to acetyl-CoA, right? We created 2 of these from pyruvate. And what's going to happen to each acetyl-CoA is it's going to enter into something known as the Krebs cycle or citric acid cycle, which is what you're looking at right here. Okay, so let's talk about the most important steps of the citric acid cycle, just like we did glycolysis. And I can already point out and tell you the citric acid cycle is or a Krebs cycle, you can call it either one, Krebs cycle. It is an oxidation machine. Okay, we're going to start ripping lots of hydrogens and consequently electrons off of acetyl-CoA throughout this process. So what's going to happen here in the first reaction is we have acetyl-CoA, and acetyl-CoA is going to combine with oxaloacetate and water in what's known as a condensation reaction. This enzyme citrate synthase is going to ultimately turn acetyl-CoA into citrate. It mostly goes from being a 2-carbon acetyl-CoA molecule to a 6-carbon citrate molecule. The next reaction, what's going to happen is citrate is just going to be scrambled a little bit into isocitrate. Isocitrate, this is an important reaction, clearly, if you can see NAD there. Isocitrate, is going to get converted into alpha-ketoglutarate by this enzyme isocitrate dehydrogenase. And in doing so, the way that this ultimately occurs is NAD comes along, steals some hydrogens off of isocitrate, and that ultimately oxidizes isocitrate to alpha-ketoglutarate, and we have now formed an NADH. But you want to remember that this reaction, right, is going to happen 2 times because we get 2 rounds of the Krebs cycle. So what we've really gained here is 2 more NADH molecules, right? 1 for each round. Next, a very similar thing is going to occur. The enzyme alpha-ketoglutarate dehydrogenase is going to assist in converting alpha-ketoglutarate into succinyl-CoA. And the way that this occurs is by NAD coming along, stealing some hydrogens off of alpha-ketoglutarate, so oxidizing it to succinyl-CoA. And what do you know? If NAD steals some hydrogens off of alpha-ketoglutarate, it becomes, of course, NADH. This happens 2 times, right? Because we get 2 rounds of this cycle. So we've just gained another 2 NADH molecules. Okay, this next reaction, the most important part of it, succinyl-CoA is going to get converted into succinate by this enzyme succinyl-CoA synthetase. And in doing so, what happens here is a phosphate is stolen off of succinyl-CoA from this molecule GDP, which is a lot like ADP. And that phosphate is taken from succinyl-CoA to form GTP, right? A lot like ATP. We've added a 3rd phosphate to GDP to make GTP. That is the way that succinyl-CoA is largely converted into succinate. And what's important to note here is that GDP is going to function a lot like kind of phosphocreatine. This phosphate, one of the phosphates from GDP, will be given to ADP to resynthesize ATP. So really, when we gain a GDP molecule from this reaction, we're actually gaining an ATP molecule. And this happens 2 times, right? So because of 2 rounds of the cycle, so we've now gained 2 ATP molecules. This next reaction, what's going to happen is succinate is going to be converted into fumarate by this enzyme succinate dehydrogenase. And this particular schematic is missing the molecule which is known as FAD. FAD is a lot like NAD. Right? It comes along, it steals hydrogens ultimately off of molecules, and oxidizes those molecules by becoming reduced to FADH. FADH2 is technically what you'll see it written as. It takes two hydrogens off of the molecule, just like NAD takes two hydrogens from the molecule. Okay, so what really happens here is FAD comes along, takes two hydrogens from succinate, and that creates fumarate. And what we get is FADH. And we get, of course, two of these. Next, what you see is fumarate, and fumarate is going, in a condensation reaction, going to be converted into malate. And then finally, malate through the enzyme malate dehydrogenase is going to be converted to one of the molecules that we started off with, which is oxaloacetate. And in converting malate to oxaloacetate by malate dehydrogenase, again, what happens here is NAD comes along, steals a couple hydrogens off of malate, so oxidizes malate to make oxaloacetate. And in doing so, we get an NADH, but we get two of these NADH molecules because, again, we get two rounds of this. So let's sort of summarize.
Before I summarize, though, I do want to point one thing out, and I don't mean to continually point things out that I'm not going to discuss in a lot of detail in the class, but I just like to do it to try to spark your interest to want to learn about this stuff more. Succinate dehydrogenase, it's what oxidized succinate to fumarate. But what's often not told to you is that you haven't seen this yet, but you're going to see something that's known as complex 2 of the electron transport chain, and that actually is this enzyme of the Krebs cycle. There's some really fascinating components to understanding that I'm not going to be, I just don't have the time to discuss in this class. But when we get to the electron transport chain, maybe you can think about that.
So let's look at the big picture of this. Okay, what happens with the citric acid or Krebs cycle is acetyl-CoA, which of course came from pyruvate. The citric acid cycle oxidizes acetyl-CoA like crazy, right? We oxidize it 1, 2, 3, 4 times as it goes around the cycle. Okay, and in doing so, right, in 3 of those oxidation steps, we gain NADH. In 1 of those oxidation steps, we gain an FADH. So ultimately, for 1 round of this cycle, we get 3 NADH and 1 FADH. And also, we get 1 high-energy phosphate off of succinyl-CoA. So we're also able to create 1 ATP for each round. You don't want to lose sight of the fact either that when we break down a glucose molecule, we get 2 pyruvates, which make 2 acetyl-CoAs, and each one is going to go around the Krebs once. So we have to double this. We actually get 6 NADH, because for each round we got 3 NADH, so times 2. We also get then 2 FADH, because each round of the Krebs cycle makes 1 FADH, and additionally we get 2 ATP, because each round makes 1 ATP. Okay, so this is what we get in its entirety from the Krebs cycle with 1 molecule of glucose. 6 NADH, 2 FADH, and 2 ATP.
And it would also be a disservice not to point out to you that carbon dioxide is created as a byproduct in multiple locations of the Krebs cycle as well, right? It was created when we turned pyruvate into acetyl-CoA, and it's also created in multiple reactions of the Krebs cycle. So when you think of that carbon dioxide that you blow off from your mouth every single time that you breathe, you can think about it as really coming from the breakdown of glucose and also fatty acids, because it's going to use this same Krebs cycle inside of your mitochondria. That's where that CO2 is actually coming from. In the blood, it ends up being quite acidic, so we constantly have to blow it off. We'll talk about that when we get to the respiratory system. But anyways, I digress.
So if we think of that glucose molecule that we broke down through glycolysis, we got a net gain of 2 ATP. If we take pyruvate, then we shuttle it into the mitochondria, we turn it into acetyl-CoA, and then we run acetyl-CoA through ultimately the citric acid cycle. We still, through all of these processes of taking pyruvate into the mitochondria, turning it into acetyl-CoA, running it through the citric acid cycle, we only gain then another 2 ATP for each round of the citric acid cycle. So far up to this point, we've gained a total of 4 ATP molecules, and that's it from breaking down this glucose molecule. So there's got to be a lot more ATP that comes from the breakdown of this glucose molecule rather than just 4. And ultimately the answer to this is all of those NADH, as well as a couple of those FADH molecules that we've created. All of those hydrogens and consequently electrons that we've stripped off of the glucose molecule are all going to participate inside of the mitochondria within what's known as the electron transport chain. So we've been stripping all those electrons and hydrogens off for a reason, and now the point is to take a look and actually see what happens with these molecules inside of the mitochondria. And that is what we will do in the next lecture.